Greetings to everyone. Welcome back once again to my educational channel on biology. Let's test for success in this video. Let's just sharpen our skills of answering exam questions and also recall what we have studied for each chapter. Today's chapter is Biology, Form 4, KSSM syllabus, Chapter 3, entitled Movement of Substances Across the Plasma Membrane. So this is the first test, is Test 3/1. There will also be another test involving essay questions, right? And we will also discuss the techniques of answering essay questions in that video. Okay, so let's begin. So for section A, there are 6 objective questions which are worth 6 marks. And for section B, there's 1 structured question worth 9 marks. So the total marks is 15 marks and the time is 20 minutes, right? So I'll read the question first and then pause for 20 seconds and then we'll discuss the answers. Now you can also pause the video after I've read the question and take your time to answer the question. The maximum time to answer each objective question is 1.5 minutes or 90 seconds. Alright, so let's start. The diagram shows the condition of some cells in a drop of fresh blood. After the blood was mixed with a saline solution, which of the saline solutions below was most probably mixed with the blood? A. 3.0% saline solution, B. 0.6% saline solution, C. 0.9% saline solution, and D. 0.2% saline solution. Okay, so time starts now, 20 seconds. Right, so the answer is A. Okay. So first of all, we have to know the isotonic solution. The solution that is isotonic to the cytoplasmic fluid of red blood cells, and that is 0.9% saline solution. Okay, this solution is isotonic to the cytoplasmic fluid of red blood cells, as we have studied, okay, in the textbook. So now we look at this picture here and we see that the cells have undergone crenation or shrinking, right? This means that water has diffused out from the cells into the solution, which is hypertonic, more concentrated than the cytoplasmic fluid of the red blood cells. Alright, so which solution here is a hypertonic solution compared to the cytoplasmic fluid of red blood cells? Is 3.0% saline solution. Any solution that has a higher concentration than 0.9% saline solution. Okay, so in a 3% saline solution, which is hypertonic to the cytoplasmic fluid of red blood cells, okay, water will diffuse out of the red blood cells by osmosis and cause the red blood cells to lose water and cause them to undergo crenation or shrinking, as we see here. The star-shaped structure here, okay, shows that they have undergone crenation. Now, for B and D, 0.6% and 0.2% saline solution, these two solutions have a lower concentration than 0.9% saline solution. So they are hypotonic solutions, alright? More dilute than the 0.9% saline solution. So they cannot cause this condition. Let's go on to question 2, which is a HOTS question. Honey is used in the healing of wounds and in food preservation. Which of the following are the reasons for this? Number 1, honey is a saturated solution of sugar which dehydrates bacteria. Number 2, honey sticks bacterial cells together. Number 3, honey has a low pH which prevents growth of bacteria. So choose the right combination of answers. Right? So what's your answer here? Now the best answer is B. Okay? So let's read the question again. Honey is used in the healing of wounds. Now, when we talk about the healing of wounds, the type of honey that should be used should be the sterile medical grade honey, not the honey that we eat, that is bought in bottles. Alright? So this type of sterile medical grade honey is usually prepared in the form of a gel and sold in the pharmacies. It's like an ointment or a gel to be applied on the wounds. Okay, so we cannot simply put any type of honey like the ones that we eat on a wound, alright, because it may not be sterile and may worsen the condition. Alright, so it's also used in food preservation. Now what's the reason for this? First choice, honey is a saturated solution of sugar which dehydrates bacteria, is correct, right? So honey forms a concentrated sugar solution which is hypertonic to bacterial cells. So water will diffuse out of bacterial cells if they come in contact with honey, okay? And the water will diffuse out by osmosis, so the bacterial cells will be dehydrated and they will die, okay? Number 2, honey sticks bacterial cells together. So this is not a reason, because although honey is sticky, it doesn't mean that it sticks the bacterial cells together, and it doesn't mean that the bacterial cells die or the growth is inhibited, even if it happens. Okay, so this is not a biological reason for the use of honey in the healing of wounds and food preservation. Number 3, honey has a low pH which prevents growth of bacteria. This is correct, because actually honey is acidic. If you have read widely or read about this, we know that honey is acidic and actually has a pH of around 3.9 to 4, and an acidic condition will prevent the growth of bacteria. Okay, so the answer is 1 and 3, B. The third question, the diagram shows the structure of the plasma membrane. Which function is incorrect for X, Y, or Z? A, Y is a glycoprotein that acts as a receptor to hormones. B, X is a carrier protein that transports glycerol. C, Z is cholesterol that strengthens the plasma membrane. And D, Z makes the plasma membrane less permeable to ions. Right? So time starts now. Right? What is your answer? Now the answer is B. X is a carrier protein that transports glycerol. Notice that the question is which function is not correct. Not correct. So B is not correct. Okay? So let's just have a look at B first. Now, X is a carrier protein. That's correct. Alright? Because the shape is like this. The carrier protein has a shape like that, so that you can change its shape and then reverse the other way, so that the opening here will be down the other side here. So it doesn't have a pore, a channel, a straight channel or tunnel here. Okay, so it's not pore protein. So it's a carrier protein that carries out facilitated diffusion, and it does not transport glycerol because glycerol can move in between the phospholipid molecules, move through the phospholipid bilayer here and move into the cells through this way, okay? Because glycerol is a lipid soluble molecule, right? Substance. So it can pass through the hydrophobic tails, all right, of the phospholipid bilayer, right? So it can pass through like this in between the phospholipid molecules, okay? So carrier protein doesn't transport glycerol, but it transports other substances like amino acids and sugar, okay? The big molecules like amino acids and sugars. Right, so how about the rest of the answers? Now, A is a glycoprotein, is correct. It has this carbohydrate chain sticking out, all right, from the protein molecule. It acts as a receptor to hormones, correct. To help the hormones move into the cell. It's a receptor to the hormones. To help the hormones move into the cells. Now, Z is cholesterol. That's correct. Cholesterol is found in between the hydrophobic tails here. Strengthens the plasma membrane, correct. And Z makes the plasma membrane less permeable to ions, is also correct. Okay. So answer is B, because glycerol is a non-polar molecule that diffuses through the phospholipid bilayer, like this way, through simple diffusion. And it does not use a carrier protein, right? So number 4, the diagram shows a liposome which can be used to transport vitamin C to body cells. In which area of the liposome is vitamin C carried? Is it R, S, or T? So find the right combination. So time starts now. Right, let's look at the answer. What is your answer? Now the answer is C, T only, in this area T. Because vitamin C is water soluble, right? So it must be carried in the aqueous solution, which is found in the center of the liposome. And that is area T, which is the hydrophilic area, the area where which is water loving, within inverted commas. Okay, because there's an aqueous solution here. So because vitamin C is water soluble, it will dissolve in the aqueous solution, right? And area S is called the hydrophobic area, in between the phospholipid tails, right? This is the hydrophobic area. R is of course out because R is on the surface of the liposome itself. So for S, the lipid soluble molecules or substances are carried here, right? And for T is the water soluble substances, carried in this part, in the center of the liposome. So vitamin C is carried in area T. Question 5. A slice of a spinach stem was immersed in 20% sucrose solution for 30 minutes. What is the expected observation? The shaded part is the epidermis. Okay, so this is the epidermis, and this white part here represents the cortex. Right, 20 seconds. Okay, so What is your answer? The answer is D. Alright, this one. Okay, so the 20% sucrose solution is hypertonic to the cell set of the spinach stem, meaning that the sucrose solution is more concentrated and has a higher concentration of dissolved substances or solutes than the cell set of the spinach stem. Thus, water will be drawn out. We say water diffuses out of the cells of the cortex by osmosis, like this. Alright. So we talk about the cortex first, and then we're going to discuss the epidermis. Alright, they have different responses. Now for the cortex cells, water diffuses out of the cells by osmosis, and the cells will shrink. So the length of this tissue, the white part here, will shrink compared to the original length, right? But for the cells in the epidermis, the shaded part here, alright, the upper part of the cells is covered by a waxy cuticle, alright, which is impermeable to water. It doesn't allow water in the cells to diffuse out by osmosis, okay? So no or less loss of water occurs from the epidermal cells by osmosis. Water doesn't diffuse out from the cells, from the epidermal cells, okay? Waxy cuticle on epidermis, water does not diffuse out of the epidermal cells. So there's a difference in the length of the cortex cells and the epidermal cells. The epidermal cells here can act as like the control. They still maintain the original length, okay, because the cells do not shrink, they do not lose water. So the length of the tissue here, the gray part here, should remain the same as before. Alright, it's like a control experiment. Okay, whereas the cells in the cortex region, the tissue has shrunk. Thus, because of the difference in length between the tissues in the cortex and the epidermal layer, thus the stem will curve. But how does it curve? It will curve in this way. Alright, now you can see here that the length of the cortex here is shorter than the length of the epidermal layer, right? So that's why it curves this way. The center part of the stem here will curve outwards, facing the outer part, because epidermal cells here are found on the surface of the stem. So this is the outer part, and the inner part is the cortex. So the stem curves outwards at the center. Some books put that the stem curves inwards because they look at the tips here. The tips curve inwards, but to be clear, we say the stem curves outwards at the center part here. It curves outwards, okay, facing the outside, because the epidermal layer is here. Alright, okay, so this is the appearance of the spinach stem in hypertonic solution. Alright, where the cells of the cortex or the tissue, the cortex tissue will shrink compared to the original length, which is represented by the epidermal layer. Okay, and for B and C, they are the expected observations for the spinach stem, okay, in hypotonic solution, more dilute solution. I can see here the cortex cells here have received water. They gain water by osmosis. Okay, water diffuses into the cells from hypotonic or dilute solution, and this length here is longer than the length of the epidermal layer here, the gray one. This one, it shows that the curvature is even more. C, for C, curvature of the stem is even more than B. So maybe this solution for C, the solution is even more dilute than the one for B. Okay, more hypotonic. And A is the observation we will get if the stem is placed in isotonic solution, which is around 5% sucrose solution. So water diffuses in and out of the cortex cells at the same rate. So the cortex cells neither gain nor lose water, and the length of the cortex tissue here remains the same as before. Okay, where like the epidermal layer, okay, which acts like a control, is the original length of the stem. Okay, it doesn't change in length. Okay, even though we have immersed the stem in whatever solution, the epidermal layer is like the control. So that's how we can understand the different observations shown here. Okay, A is for isotonic solution, B for hypotonic solution, and C is when the stem, D is when the stem is placed in hypertonic solution. Okay? B and C is for hypotonic solution. Question 6. Which of the following processes is slowed down by a lack of oxygen or by a respiratory poison? A. Absorption of water by root hair cells. B. Gaseous exchange in lungs. C. Absorption of fructose molecule in the villus. And D. Absorption of mineral ions by root hair cells. Okay, so the answer is D. Absorption of mineral ions by root hair cells. Okay, so let's read the question again. Which of the following processes is slowed down, becomes slower? Caused by a lack of oxygen or a respiratory poison? Either one. So when we look at this question, it mentions a lack of oxygen or respiratory poison. We know it has something to do with production of energy through cellular respiration. Okay, so it will affect, this lack of oxygen or respiratory poison will affect the process of active transport. Alright, such as the absorption of mineral ions by root hair cells. It's by active transport. So active transport requires energy, right? And when there's a lack of oxygen or respiratory poison in the cell, alright, it slows down cellular respiration. A respiratory poison is a substance that will slow down cellular respiration in a cell. So it's dangerous for the cell. And a lack of oxygen also causes cellular respiration to slow down, because oxygen is needed for cellular respiration. According to the equation GO-WEC, glucose plus oxygen produces water, energy, and carbon dioxide, right? So a lack of oxygen or respiratory poison slows down cellular respiration and the production of energy through cellular respiration. That means the cell will get less energy already for active transport. So this will slow down active transport of substances such as absorption of mineral ions by root hair cells. Okay. Now for process A, B, and C, they all involve passive transport which does not require energy. Okay, let's go through them one by one. Absorption of water by root hair cells is by what process? Osmosis, right? Gaseous exchange in lungs by diffusion. Absorption of fructose molecule in the villus by facilitated diffusion. Alright, so these three processes, ABC, are processes that involve passive transport. So remember the four types of movement across the plasma membrane is SOFA, S-O-F-A. S for simple diffusion, O osmosis, F is facilitated diffusion, and A is active transport. Alright, and here's a picture to show you the root hair cell with this projection called the root hair. So we have to be aware that water diffuses into the root hair cells by osmosis, a type of passive transport. But mineral ions are actively taken up by the cell. That means mineral ions move into the cells by active transport, where the cell needs to produce energy for this process. And the example of mineral ions are magnesium ions, nitric ions, calcium ions, etc. Class, so now we'll look at the structured questions, right? There's only one structured question. It's worth 9 marks, and the time for it is 12 minutes. So you can set the timer in your phone to answer this question afterwards, right? So you can pause the video at this point and then take out a piece of paper and try the question yourself and write down the answer, right? And afterwards, when we discuss the video, you can mark your answer yourself. In that way, you will know how marks are given to, right? And you'll be better able to answer the question from the viewpoint of the examiner, right? So you'll be able to answer the question better. Okay, so you can pause the video here, and then I will go on to the next slide after this, which has the answers for this question. So try the question yourself in order to let your brain muscle, your brain in inverted commas muscle, the brain muscles exercise, because the brain is like a real muscle. The more you exercise it, you train it, it will learn, and you will be able to answer, give better answers in the exam, in the real exam. Also, we'll learn the technique of answering these questions. All right, you can pause the video now. Right, let's look at the answers. So diagram 1 above shows different types of movement of substances across the plasma membrane. Now here we can see two types of movement of substances, right? The first one involves molecule H that moves through the phospholipid bilayer, right? And the second one involves molecules F and G, which carry out the same process as stated here. All right, so they carry out a type of movement of substances across the plasma membrane. All right, so in diagram 1, circle a phospholipid molecule and label it P. Right, so follow the instructions. Circle a phospholipid molecule here, which has a hydrophilic head and 2 hydrophobic tails. Right, then you must label it P. Without the label P, you will not get the mark. So you can also circle the phospholipid molecule below here. Okay, any one way, and use a pencil to circle the molecule. Next, let's go on to question B. Molecules F and G carry out the same process. Name the process of movement of molecules by F and G. Okay, so first of all, we have to determine what this molecule F and G is. It is a carrier protein, right? And then is it active transport or passive transport? So there is no ATP involved, right? If there is a symbol showing you that ATP is involved, then it is active transport. Okay, but there's no symbol showing the molecule ATP, right? On top of that, we can see a lot of these molecules outside. So the concentration of the molecules is higher outside the cell than inside the cell. And the carrier protein F and G, carrier proteins F and G are transporting the molecules from region of higher concentration outside to region of lower concentration inside the cell. So it is a form of passive transport. Okay, and there are only 3 types of passive transport, right? And the one that involves the 3 types of passive transport are simple diffusion, osmosis, and facilitated diffusion. Okay, the one involving the carrier protein must be facilitated diffusion. So the answer is facilitated diffusion. Now, if you write facilitated transport, you'll get it wrong because the name is facilitated diffusion. It is a type of diffusion. All right. Now, C. Explain how amino acid molecules are transported across the plasma membrane by F. All right. So highlight amino acid molecules, F, which is a carrier protein. Okay, so that means you have to explain the process of facilitated diffusion. All right. So 3 marks here. Now, first we must state that the amino acid molecule binds to the specific site on the carrier protein here, right? Next, the carrier protein changes shape from shape in F to the shape of G, from shape of F to the shape of G, where it opens up. At first, for F, it opens up to the environment, right, to get the molecule. And then after that, it changes shape so that the carrier protein opens up to the interior or the inside of the cell so that the molecule can enter the cell. Okay, so the carrier protein changes shape to allow the amino acid molecule to pass through and enter the cell, right? That's point number 2, right? And then the carrier protein returns to its original shape. It will go back to the shape like in F and is ready to transport other molecules down the concentration gradient. So keywords here are number 1, binds to the specific site, okay? And then number 2, carrier protein changes shape, allow amino acid molecule to enter the cell. And the third point is carrier protein returns to its original shape. Down the concentration gradient, there may be one more mark for that. Or you can write transport molecules from region of higher concentration outside the cell to region of lower concentration in the cell. All right? So remember the keywords, all right, when you write the answer. Okay, next, D. State a characteristic of molecule H. Molecule H here, that enables it to pass through the plasma membrane by the method shown. That means through the phospholipid bilayer, through the pores found in the phospholipid bilayer. Okay, so if it can go past the phospholipid bilayer like this, it may be lipid soluble, or it may be a molecule that's very small in size, like water. It's very small in size, so it can go through the pores in between the phospholipid molecules. All right. So it's lipid soluble, or you can write it is small in size, like water, like for water. Now, question E. So for D, highlight the word characteristic. They want the characteristic of the molecule H. Now for E, a chef prepared a vegetable salad and mixed some thick sweet sauce with the salad. After 20 minutes, the thick sauce became dilute and watery. The vegetables became soft. State the process that occurred in the cell, 1 mark. Okay, so read the question a few times and try to find out what actually happened to the vegetables. The vegetables became soft. So there is something that tells you that osmosis has occurred, and when it becomes soft, it means that it's probably lost water, right? So water is diffused out of the cells into the thick sauce. That's why by osmosis, water is diffused out of the cells of the vegetables by osmosis, and it made the sauce watery. Okay, think of rojak, our local fruit and vegetable salad. Rojak, this is what happens to the rojak if you leave it for some time on the table without eating it. The sauce will become dilute and watery, and the vegetables will become soft, like this. Okay, so that means the thick sauce is the hypertonic solution, forms a hypertonic solution to the cell set of the vegetables. Then water diffuses out of the vegetable cells by osmosis, and this makes the vegetables lose water, so the cells become plasmolyzed, and also they cannot provide support to there's no support for the cells, all right, or for the tissues anymore. So the vegetables become soft. Okay, now state the process that occurred in the cell. Plasmolysis is the answer. Okay, now not osmosis, because osmosis leads to the process of plasmolysis, and that's the process that occurred in the cell. Plasmolysis means the shrinking of the cytoplasm and the vacuoles when water diffuses out of the plant cells by osmosis. When the plant cell is placed in a hypertonic, a more concentrated solution, a hypertonic solution. Let's go on to the last question. The last question. Draw one vegetable cell to show its condition after it has been immersed in the thick sauce, right? So since the cell is plasmolyzed, we have to draw a plasmolyzed cell. So here we have drawn the cell. The cell wall is still intact and retains its shape because it's rigid, right? But inside, the cytoplasm has shrunk and the vacuole also shrinks, right? So the plasma membrane here will pull away from the cell wall. It's no longer attached or stuck to the cell wall. So the plasma membrane is here, and this is the cytoplasm that has shrunk, right? Okay, so marking scheme is like that. If your drawing is correct, showing a plasmolyzed cell, right? And then you must show the vacuole being smaller than normal. Don't draw a big vacuole. And then because the vacuole has shrunk, plasma membrane pulls away from the cell wall. Must be like this. Cannot draw a plasma membrane still stuck to the cell wall. No broken lines for the diagram. It must be neat, and there must be no shading like artistic touches to the to the cell. There's no need to shade any part of the cell. Alright, so that's 1 mark for the drawing if the examiner is satisfied with your drawing. And then 1 mark for the label. So you must have at least 2 labels. Each label is like half a mark. But we don't give half marks, okay, in the exam. So you must have 2 labels to get 1 mark. So even if the diagram doesn't ask you to label, please label the diagram. It's understood that when you draw a diagram, you should label it so that people understand what you have drawn. Okay? So label the parts that are important. Now, if you label the nucleus, you may still get your mark. But important parts are cytoplasm, plasma membrane, vacuole, and cell wall, to show the effect of these osmosis on these parts. Okay? Right. So that's all for this lesson. Thanks for watching. Thanks for trying out this question. I believe that you will benefit from it if you keep testing yourself and also marking your own paper. Okay? In order to know the marking scheme too. How is it marked in exams? How are the questions marked? And also in order for you to know the keywords. So how did you fare in this test? Now, some students may find it a bit challenging, that this test is a bit challenging, because there are certain hot questions there. However, it's good for us to try this type of questions so that we are better prepared for difficult questions in the exam, right? So here's the score chart. If you have a score of 14 to 15 marks, it is equals to 93 to 100%, and this is an excellent achievement, and the grade is A+. 12 to 13 marks is equals to 80 to 87%. Very good achievement, worth an A, right? And then 10 to 11 marks, percentage is 67 to 73%, moderately good achievement, B. 8 to 9 marks, percentage is 53 to 60%, moderate or average achievement, C. 6 to 7 marks equals to 40 to 47%. Results can be improved, don't give up. Try more questions, memorize the facts well, understand the concepts too, right? And learn from mistakes. Same for those who have scored 5 and below, is equals to 33% and below. So revise your facts, try more exercises, keep on learning, and learn from mistakes too. Alright, so that's all for this lesson. Goodbye for now.